Hello, my seniors. Welcome back from break. We're going to get started here talking about topic 3.1 and 3.2, and they focus on genes and chromosomes. Let's get started. So a point to ponder right from the beginning is, what is the difference between a gene and an allele? So keep that in mind. I know we've talked about this before in 10th grade, but they're two words that often get said at the same time, and sometimes people think they're the same thing. So keep that in mind as we go through this lecture. All right, so we always want to keep the big picture in mind. So where we just came from in our last unit was we were talking about DNA and how it turns into proteins. So when we talk about different vocabulary in this unit, you're going to hear a lot of the same terms. So I want to kind of go from the small end of this picture to the big end. So if you look down here, this bottom word nucleotide, remember this is a picture of a nucleotide, one phosphate, one sugar, and a base. If you put a bunch of nucleotides together, you're going to get a strand of DNA. On this DNA are genes, so different pieces of the DNA are representing your genes and your alleles, which are different options of those genes, like the gene for blue eyes, and then the allele could be brown eyes, or blue eyes, or green eyes, or hazel eyes, but they're the same gene technically. When the DNA is all coiled up, it creates chromosomes, okay? And so on chromosomes, since they're made of the DNA, they have the genes. And then you can find these chromosomes, or our DNA, in the nucleus of the cell, and our cells are eukaryotic cells. So just to kind of review the vocabulary that you're going to hear again from the last unit. Okay, so here are we're going to go through 2 slides of some terms that are easily confused. And you want to make sure you're clear on these definitions. So DNA. DNA is our double-stranded helical nucleic acid molecule containing genetic instructions. So to break that down, double-stranded, it's in a helical shape. So in this picture here, you can see it's double-stranded and it's twisted in a helical shape. And it's made of nucleic acids, right? Or it's made of nucleotides, which then when all put together, we call that a nucleic acid. So remember, there's 2 nucleic acids we've talked about, DNA and RNA, hence the Na for nucleic acid. And it contains our genetic instructions, right? What proteins are going to be made. Chromosomes are only in eukaryotic cells, and we'll go over that more later, but just remember that. The structure of a chromosome can only be found in a eukaryotic cell. And just to get an idea of what a chromosome is it's a tightly wound DNA around histone proteins, and you get something that looks like this image right here. The green is the histone proteins, which gives it structure, and then the DNA is wrapped around it. And it's also in a helical shape already, right? It's double-stranded, so it's extra helical. So DNA plus proteins, histone proteins, is what's going to create a chromosome. Genome. Genome is one we're going to use this unit. We haven't really used before, but the genome is the whole of the genetic information of an organism. So we would normally say your genome size is based on the amount of DNA in your body or in an organism specific to that species. So, for example, the Human Genome Project is an internationally worked on project where we sequenced what each part of our DNA codes for, what gene they code for. So we know what each gene that humans could possibly have. They've done this sequencing for other organisms as well, but the Human Genome Project was huge in the idea that we could research and figure out maybe what's causing different genetic diseases, where the mutations lie in those genes to cause those genetic diseases for some individuals, and it helped us really understand ourselves more as a whole, as a species. Gene is a heritable factor that consists of a length of DNA on a specific chromosome and controls a specific characteristic. So, for example, in this picture here, this is a picture of 2 homologous chromosomes. So they each carry the same genes, but possibly different alleles. But you can see here that this section of DNA that's wrapped around this chromosome is for gene 1, and this section of DNA that's wrapped around this chromosome as well, probably lower than gene 1, is gene 2. So they both code for a different trait in your body. An allele would be a specific form of a gene. So it's different from other alleles by usually only a few bases. So for example, when I say bases, I'm talking about A, G, or A, T, Gs, and Cs. So that means that maybe you have one allele for brown eyes and one allele for blue eyes, right? And one of them might be more dominant. But they're only different by a few of those base pairs. So maybe one has 3 Gs and the other one has 2 Cs and an A. So just a few little bases make these options different. And now we have different varieties of certain traits or certain genes, and we call those alleles. So just to give you a visual of, here's a chromosome and where we would find the loci or location of a gene. So this is one gene or one chromosome, and this would be a gene here, and this would be a separate gene down here in pink. So this gene, to simplify it, would be eye color, and this would be the DNA, the piece of DNA wrapped around in this part of the chromosome. And this base sequence codes for this person's allele, which would be, or trait, and that would be they have brown eyes. For this pink one, we have hair color, so this is the DNA wrapped around here, and apparently this person's hair color is hazel. I've never heard someone's hair color be called hazel, but this picture says it. Also, when we talk about chromosomes and we talk about where certain traits are on this chromosome, there's a certain kind of language that you would need to know if you studied genetics and you studied chromosomes. So, for example, this is chromosome 7. So we have 23 pairs of chromosomes. So this is one of the pair of chromosome 7. This is called the centromere right here, where it pinches together. And we call the top section P, and we call the lower section Q. So the top section, the short arm, is represented by P, and the bottom section, or the long arm, is represented by Q. Okay, so if you look over here on this chromosomal location code so far, we have chromosome 7. The gene we're going to be talking about is in the Q section of the chromosome, so that must mean it's on the long arm. This region right here, so they break up the chromosome into regions. So this region is called region 3. And this is band number 1 in this blue box right here. And then this other arrow right here is talking about this gray band, and that's called subband 2. So this band is representing a location of where a certain sequence of DNA is on that, and that would be called a gene. So we would call this chromosome 7, or this gene is located on chromosome 7 and the Q band in the region 3, in one band in subband 2. So this is how, and notice the decimal is there to represent that it's a subband. So this is how people talk about chromosomes. I'm not going to test you on this, but I want you to, if you're interested, we want to go a little deeper here, and this is where you would find the gene for CFTR. All right, so just to be clear, like I said before, eukaryotic cells have chromosomes and prokaryotic cells don't. So it's because in a prokaryotic cell, in this image here you can see, they only have one circular chromosome. And here you're going to see that they also contain plasmids, which are circular sections of DNA, usually small sections. But usually there's just one circular section of DNA, and it can be found in the nucleoid region. For eukaryotes, we have linear chromosomes that are associated with histone proteins because they're DNA wrapped around histone proteins. Different chromosomes carry different genes. The number of chromosomes is a specific characteristic of a species. So for example, we have 46 chromosomes, 23 pairs, while a horse might have 26 chromosomes, which would only be 13 pairs. Okay? So that's something that separates a species. It's also a big factor on why random different species can't mate together, okay? Because the chromosomes won't add up properly and they don't match. Okay? So realize that the number of chromosomes is very specific to each species, but only eukaryotic cells or species with eukaryotic cells have chromosomes. Okay, so let's focus on us, chromosomes in humans. So when we talk about chromosomes, we always talk about, and you might remember these terms from 10th grade, we have diploid cells. So diploid cells means that each cell in our body has 2 sets of each chromosome. So we usually define that as 2n. So 2 times n would be the amount of chromosomes if it was haploid. So in our sex cells, we refer to it as haploid cells, and that would only be 1 set of each chromosome. So it's half the amount. Remember, because half of your information came from your mother and half came from your father, right? So 23 of the chromosomes came from your mom and 23 came from your dad, and they mixed together and they created a unique combination of you during meiosis. So we say 2 times whatever n is going to create our amount of chromosomes in an individual. So that would be 46 chromosomes. So if you look here in this picture, you'll see 23 pairs, but technically 46 chromosomes. We also refer to chromosomes as being called homologous chromosomes. So homo means the same, so we match them up in groups, meaning that they carry the same genes on them. So both these chromosomes, don't worry about the structure or the shape, have the same genes on them, right? So on this chromosome, you're going to have 1 allele for eye color for that gene, and on this chromosome, you're going to have another allele for eye color on it. So you might have 2 of the same color alleles, 2 different ones. It just depends on which is dominant that you're going to express in your characteristics. But this is a homologous pair because each of the chromosomes carry the same gene on them. Now, we call the first 22 pairs autosomal chromosomes or autosome chromosomes, and they are the same in males and females. So they each carry the same genes on them. So your pair 15 of chromosome is the same as my pair 15. They have, whether you're male or female, they carry the same genes and they have the same effect on a human's body. The only one that would be different is the last one here, the 23rd pair, which is your sex chromosomes. So this is a female because they're both X chromosomes. We call this here an image a karyogram, and this is how we can study for different diseases or just understand the sex of an individual. Now, chromosomes are not floating around in your nucleus like this image. I'm going to show you later how we do this, but they are usually, it's more of a process than this. Someone has cut the image up and matched them and figured out this karyogram or this sequence for this individual. Here's just to give you another visual of homologous pairs. So this is a homologous pair and this is a homologous pair. So this would be chromosome pair 1. They each have the same gene on them. 1 came from the mom, 1 came from the dad, okay? And this would be your gene here, maybe let's say for eye color, this band up here, and this would be one version of that eye color, and this would be another version of that eye color. This would be chromosome pair 2, it's a homologous pair, and they carry the same genes on them as well. So one thing we have to be clear about is different numbers of genes for different species is important. The number of genes does not necessarily correlate to the complexity of an organism. So they're all a little bit different. Like, for example, here you see this is the influenza virus on the lower portion, and it only has 11 genes as opposed to a grape, which has 30,434 genes that it can express, where a human has 22,333. Now, I would think we're more complex than a grape, but they have more genes that could potentially be created. Now, when you think of genes, it doesn't always mean like brain function. It could be proteins that their bodies or structures create. It's not something maybe like color of the skin of the grape or seeds versus the size of the seeds, I guess we could say. So it just depends, and it doesn't always mean like it's smart, right? So the number of genes does not necessarily correlate to complexity of an organism. Also, when you look here, the number of chromosomes does not always correlate to the complexity of an organism. So the number of chromosomes is the diploid number in green. So if you look here, the dog actually has more chromosomes than us, okay? Or not chromosomes, but yeah, chromosomes. We have 46. And here, I'm not exactly sure because the number doesn't line up, but I'm going to guess it's somewhere in the 80s. And you would think that that means they're more complex, but it's not. It does not mean that. And then the number of genes is in pink. So like we talked about here, here's rice. Look how many genes they have, different varieties of genes. That's a lot versus humans over here. And then the size of our genome. So that means the amount of DNA in an organism. So we have a lot of DNA here. So that was a lot that they sequenced compared to, for example, a fruit fly or a bacteria. Okay, so this is how we determine chromosome size, and it's a technique called the Karen's technique or Karen's technique. I'm going to go through it using a picture because I think it's better, but here are the steps for you. This has been a question in the past on IB exams explaining the steps, so here it is for you here, but I'm going to use the picture to help. All right, so here's the picture. So if you look through it, it flows this way and down and zigzags through. So you first prepare the DNA. So you're going to incubate the cells in a radioactive thymine, and you're going to gently lyse those cells so that radioactive thymine can attach itself to the DNA. You're going to bed the contents onto a slide. You then immerse the slide in a radioactive sensitive emulsion. It's kind of like when you do old photography, when you're trying to get the prints ready. Then you dip the slide in silver bromide emulsion, and that's going to, the radiation then activates nearby the silver ions, and then the activated silver ions are reduced to silver metal, and then you're going to have a soluble ions washed away, and that's going to leave that silver metal on the picture, and that creates an image. So you're going to expose the slide to photographic film, and the develop then will transfer to image film, and then you can look at it through an audio radiograph. And at the end of it, what you're going to get is something like this here. So this is what the DNA actually looks like afterwards, because it's usually all coiled up. It's not in the X formation like it would be during mitosis or meiosis. And then someone goes and they cut through the DNA and they match it up by size. So you're going to end up with a karyogram down here like this. What's interesting is this picture is during the moment when they had gotten this DNA, that DNA was going through transcription. So you can see where it was cut here, and it was actually transcribing a portion of this into mRNA, probably. So it's open in this location. Okay, so a karyogram is the organization of chromosomes of a cell according to size and structure. So you can see here we have chromosome 1, chromosome 2, chromosome 3, and it usually goes from longest to shortest, you can notice, except for the sex chromosome. So these ones are both longer, and these ones, for example, are very short. So each human cell has 23 pairs of homologous chromosomes. Remember, each of these are homologous. One came from the mom, one came from the dad, and they have the same genes on them. 22 of the pairs are autosomes, and 1 pair is a sex chromosome. Now, what's great about karyograms is they use these to detect gene mutations. So this would be a change in a base sequence of a gene. New alleles are formed from gene mutations, so they can use that to look at that, but they can also use it to look at sex determination. So if you look here at this picture, which is male and which is a female. So a male is an XY chromosome and a female is an XX. So if you look down here at where the sex chromosome usually is in a karyotype or a karyogram, because it's the image here, think of it like a photogram or photograph. It's the karyogram. This would be a female because they're 2 long chromosomes, so that means they have 2 X chromosomes. And this is an XY because the Y is always very short. Also, we can look at it to look for different genetic diseases that have occurred. For example, if someone inherits an extra chromosome, like this one here, chromosome 21, this would be when they have trisomy, is what we call that, because it's 3 chromosomes, and this one causes particularly Down syndrome. People can also inherit an extra X chromosome or an extra Y. There's a few places in here, and we're going to go through them in another activity, where you can inherit an extra chromosome, and sometimes they and usually can be pretty fatal or very much affect development of a human. So just to talk a little bit about Down syndrome, so this is trisomy 21 because it's on the 21st homologous pair and you have 3 chromosomes. You inherited an extra copy of the chromosome. So that would be an accident happening during meiosis. So the diagnosis and the way you can usually tell is there's a single crest or crease across the palm of a hand. There's also a slightly flattened facial profile and an upward slant to the eyes. And usually what comes with it is developmental disabilities as well. Another inherited disease, since we're talking about chromosomes and DNA, is the form of anemia. And it's a condition in which there aren't enough healthy red blood cells to carry adequate oxygen throughout your body. So if you look here in the picture, your hemoglobin or red blood cells, which are proteins, remember, should look like this so that they can hold a lot of oxygen. They should be in a circular shape. But what happens is there's a mutation and it forms something called sickle cell or sickle cell anemia. And they turn into this half moon shape or these tiny slivers, so they can't hold as much oxygen. So you then have anemia. Your blood can't carry enough oxygen through the body. And the reason this happens is there's a DNA mutation, which is a base substitution. So just one letter shifts. And this causes a change to the base sequence then of the mRNA that's transcribed, which then would cause a change to the sequence of the polypeptide protein, or hemoglobin in this case. So it's now the wrong shape. So the wrong structure is going to make the wrong function, and you're going to end up with an inadequate blood cell. So the result is this causes glutamatic acid, which is glue. This is one. Remember, we have 20 amino acids, and glue is one of them, to be substituted by valine, which is another amino acid. So now instead, glue becomes val, and it's the 6th amino acid in a hemoglobin polypeptide chain, or protein. So the 6th amino acid is always the one that's changed, and that creates this shape if someone has or inherits sickle cell anemia. So blood is produced in the thin sickle cell shape, which they call like a crescent moon shape, and it can't hold as much oxygen. So one interesting thing, though, just as a side note for talking about sickle cell cases, sickle cell is a genetic trait. It's more commonly found in Africa. But what they've found is that people who have sickle cell, or at least they are carriers of sickle cell, not someone who's completely has sickle cell, because sickle cell is recessive. So if you have both alleles on that homologous pair are for sickle cell, then you're going to express it, and you're going to have sickle cell anemia, and it's deadly eventually. But if you are a carrier of it, so meaning you have the dominant trait to have normal blood cells, but you are carrying the recessive trait, you are actually resistant to malaria. And if you notice, in Africa, which has the largest cases still of malaria, though it is decreasing, they have become more resistant to it because many people are sickle cell carriers, and not that they have it, but they are carriers of it. So that means they are going to be resistant to malaria, and malaria is a big issue in Africa. So in a sense, this says it has an adaptability function as well. As we move into evolution, eventually you'll hear about this again. But that's kind of an interesting connection between sickle cell anemia and adaptations. Okay, we'll be doing this karyotype activity in another class. I'll be telling you about it. But please put your notes into ManageBack for me, and please make sure you've listed any questions you have, and we can talk about them at the beginning of next class. Let me know if you have any questions, and I hope that was helpful.